All right, so we finished a conversation about the heart itself. Now we're moving towards the discussion of blood vessels and regulation of blood pressure. So first we're going to chat about different types of blood vessels. So we've got three main types: arteries, the arterial wall is shown here. They're pretty thick. They carry the blood away from the heart, always. So those are reds. Our arteries here, in blue, pulmonary arteries. Veins always carry blood towards the heart. It's pulmonary veins and veins of the systemic circuit. Then there are capillaries that are basically in between, so blood goes in the following pathway. It goes through the arteries, through the capillaries, through the veins, back to the heart. Okay? So arteries, capillaries, veins. Now, both arteries and veins have three distinct layers, three distinct tunics. Tunica intima is the innermost layer that comprises endothelium. Endothelial cells are essentially simple squamous epithelium cells lining the inner surface of the blood vessels. And basal lamina, basement membrane, that attaches cells of tunica intima to the tunica media, which is composed mainly of the smooth muscle cell. You can see that smooth muscle layer, tunica media, is much thicker. Hold on a second, I'm going to dial down the light. So tunica media is much thicker in arteries than in veins, which explains why arteries have much more pronounced vasomotor response. They can dilate and constrict. And then on the outside is tunica externa, that contains a lot of collagen fibers for protection, elasticity. In some larger vessels can contain so-called vasa vasorum. Vasa vasorum shown here is the cross-section of blood vessels. So vasa vasorum, here, vasa vasorum, is the blood supply for the vessel wall itself. The micrograph in the left bottom corner of the slide shows you the cross-section, the vein, the artery. You can see that artery is much more, I would say, composed. It's more circular. And usually arterial lumen, generally speaking, is smaller than the lumen of a vein. Artery is more constrictable. Because of that thick, smooth muscle layer, it better keeps its circular shape. Vein is much more flat, can be flattened out. Does that make sense? So capillaries have only a layer of endothelium and basal lamina. So they don't have tunica media or tunica externa. We generally say they don't have any tunics. And that very thin layer, just endothelial cells and basal lamina, allows a rapid exchange of gases and nutrients between the blood in the capillaries and the tissue. There are three distinct types of arteries. First type is elastic, shown here on the left. Those are the largest arteries in the circulation, such as aorta, subclavian arteries, common carotid arteries. You see that they have really thick tunica media. However, tunica media is not entirely composed of smooth muscles. It also contains a considerable proportion of elastic connective tissue, which makes these arteries, well, elastic. They can easily expand and recoil in response to changing blood pressure, which explains their main function to buffer changing blood pressure. Because if you think about it, your normal systolic blood pressure is 120, normal diastolic is 80, and the function of these blood vessels is to buffer these changes, making the blood flow through the smaller vessels smooth. Are we clear? These vessels have pretty low resistance due to a large diameter, and very importantly, they have practically no vasomotor responses. Do you understand the word vasomotor response? Vasomotor refers to the ability of the blood vessels to constrict or dilate. And it makes perfect sense that the largest arteries can neither constrict nor dilate. Think about the possible consequences of common carotid artery constriction. It will basically shut down the blood flow to half of your brain. Does that make sense? So yeah, no vasomotor response. Now the smaller arteries, smaller in diameter, are called muscular arteries. They have much higher proportion of the smooth muscle. They have extremely active vasomotor responses, and their main function is to regulate the blood flow to the organs. Is that understood? So for instance, arteries that, renal arteries, can constrict and dilate, regulating the blood flow to the kidneys. Are we clear? Now the third type, the smallest arteries, are called arterioles. They're shown here on the right. They have thick tunica media with a good proportion of smooth muscle. They are small, about 30 to 50 micrometers in diameter. 37 is sort of an average size, but size varies. So these arteries are also called resistant vessels. Arterioles are perhaps the most important blood vessels that regulate the systemic blood pressure. Their function is to deliver blood to tissues, and therefore they can regulate the tissue perfusion. You may ask, what's the difference between, say, muscular arteries and arterioles if they both deliver blood? Well, consider this. Say, liver is quite large. So, muscular arteries, hepatic arteries, will regulate blood flow to the entire liver. But inside of the liver, it can be normal, constant imbalances in the blood flow. So the parts of the liver that require increased blood flow, this increased blood flow will be provided by vasodilation of the arterioles. So basically, arterioles regulate blood supply to small patches of tissue. Does that make sense? Do you understand the difference? We're going to come back. I will ask you, basically match the definition and the term. Now, veins can be divided into three categories as well. Venules, also called postcapillary venules, are the smallest type of veins that collect the blood from capillaries. You can see that the main type of tissue found in those postcapillary venules is the connective tissue, to ensure that they not losing any integrity. The proportion of smooth muscle is fairly small. Veins do not have active vasomotor response. Does that make sense so far? They do not have active vasomotor response. They do not really constrict or dilate. They function it's kind of a site of exchange of cells between the blood and the tissue. For instance, in case of infection, white blood cells can move from the bloodstream into the infected tissue. Are you with me? And that movement occurs in postcapillary venules. Okay. Now, as venules converge, kind of many tributaries make a large vein, become medium and large, and as the size of the vein increases, the proportion of the smooth muscle increases as well. Now, larger veins have fairly thin walls compared to the lumen diameter. So that's the venular lumen, and that's the wall of the vein. So they often are called capacitance vessels. Now, if you think about it, you have about 5 liters of blood circulating through, and we're going to focus on the systemic circuit, okay? Circulating through your blood vessels in the heart. Does that make sense? And we mentioned that actually stroke volume isn't that large. We're talking about, 100 mils or something. So where's all the blood? Turns out that all the blood, not all the blood, 2/3 of human blood is in veins. Veins are stretchable. We call them capacitance vessels. How to compare them? Well, think about this. Think about the river that runs from the hill into the valley. As it runs downhill in the fairly narrow riverbed, the stream is fast. Do you understand what I'm saying? Like a mountain river. But when it enters the valley and the bed becomes wider, the flow, the velocity of the flow goes down. We're clear? So that's basically the idea of capacitance vessel. Now, sorry. Third specific type of veins that we mentioned in anatomy, venous sinuses. They are flat with very thin walls, found in the cerebral circulation and in the coronary circuit. Okay. So you see that mostly the biggest difference is between the small postcapillary venules and medium and large sized vessels. Good? Any questions? Capillaries. Now, capillaries, as I mentioned, consist of a layer of endothelial cells and the layer of basement membrane or basal lamina. Okay? In some cases, there are other cells called pericytes that would be distributed on the surface of the capillary. So pericytes would control capillary permeability. Okay? How big they are. Capillaries are the size of a single cell. If you would watch the movement of cells through the capillaries, it's not free. I mean, it's not a smooth fast flow. Blood actually flows very slowly through the capillaries, and on top of that, you look at the cells, like white blood cells, they quite literally are squeezing through. Does that make sense? Now, blood vessels are not found in some tissues, like epithelial tissue is not vascularized at all. Cornea is not vascularized. Eye lens is not vascularized. Cartilage is no vascular supply. Okay. Now, the thin wall of the capillary provides a really quick exchange of nutrients and respiratory gases between the blood inside of the capillary and the tissue outside. Does that make sense so far? We're good? Okay. So there are, in fact, if you would look at the human body, every single cell in the human body, I'm not talking about like the blood vessels themselves, but like tissues, tissues that are external to blood vessels. Every single cell is located 50 or less micrometers away from a capillary. Does that make sense? So there is not a single cell that is farther than 50 micrometers from the capillary, because that's the distance that diffusion still goes over effectively. There are three types of capillaries that you must know. One is continuous. That's the least permeable. These capillaries can be found in the skin, which makes sense. You don't have really a lot of exchange in the skin. It's also, if you think about it, it's a safety measure. Your skin is exposed to all kinds of stuff. Does that make sense? So, I don't know, you are exposing your skin to pathogens. You don't want pathogens to be able to get into those capillaries. Muscles and brain, especially in the brain. It's really important. Tight junctions between endothelial cells keep blood in the capillaries and the tissue well separated. So only nutrients and respiratory gases will move between the blood and the extracellular interstitial fluid in a very controlled manner. More permeable type is fenestrated capillaries. Those little dots in the endothelial cells are called fenestrations, from the Latin fenestra. I don't know the proper, I know that in French it's fenestra. It means window. Okay, so those tiny little fenestrations, windows, provide a patent way to larger molecules, such as proteins, for instance, or larger chemicals to go from the blood into the interstitial fluid and back. Okay? These capillaries are very suitable for filtration or absorption. You can find them in the tissues like kidneys, where filtration and absorption is all that they do, really, or capillaries that run through the villi, the small intestine, that absorb nutrients. Finally, the most permeable type of capillaries is sinusoid ones. It can be found in the tissues where there is an intensive movement of even cells, not only protein cells, between the blood and the interstitial fluid. Liver, that's your metabolic center. Bone marrow, blood generation. Spleen, sequestration of the old blood cells and getting rid of the pathogens. Adrenal medulla, response by releasing hormones. These cells are poorly connected to each other. You can see there are huge intercellular gaps. Usually it's not shown on the picture. The cells are usually fenestrated as well. And the blood flow in sinusoids in these most permeable capillaries is pretty slow. That slow blood flow allows for the quick and efficient exchange between the blood and interstitial fluid. However, you can imagine that being so permeable, the sinusoids are the great site for various pathogens to go from the blood to the tissue. This is why in the sinusoids, in basically the outer lining here, you're going to find a bunch of macrophages, such as Kupffer cells in the liver. Any questions? Catch-ups? Good? So, now, the word capillary bed. It refers to a specific set of microvessels that supply different parts of tissue. So this is a capillary bed. Now, if you would look at the boxes with blood vessels right there in the corner, you will see large vessels, right? Big arteries. If you ever have a chance to visit an exhibition that is called something like Body Revealed, or you know what I'm talking about? Yeah, the plastified humans. Depends on the company that runs this exhibition. It's totally worth the money, believe me. Yeah. I happened to see a plastified blood vessel network of a human. So what they've done, basically filled all the blood vessels, including the smallest ones, with some kind of polymer, and removed the rest of the human. Well, guess what? You basically have the human. There are so many blood vessels that you can see the shape of a person, facial features, all of that. Microcirculation, the blood vessels that generally considered to be smaller than 100 micrometers in diameter, represents 95% of all blood vessels. To give an idea how important the microcirculation is, the total surface of microvessels is roughly comparable to a surface of a football field. And that is all site of exchange. So, what forms microcirculation? Small arterioles, like this arteriole, like this met arteriole. Small veins, like thoroughfare channel, or post-capillary venules. And of course, capillaries. The importance of microcirculation is greatly appreciated now. It turns out the first signs of cardiovascular disease appear first in the microvasculature. Now that kind of vague and dry statement means the following. If you have a patient who you suspect is at the risk of developing cardiovascular disease, you can assess microvascular responses. You can't do it. There are methods in humans. And you can see how this person's microvascular responses compare to sort of general, to the normal ones. Does that make sense? And you can say, okay, hey Philip, although you don't have any atherosclerosis or angina pectoris, or you have minimal atherosclerosis, you have endothelial dysfunction, you have microvascular dysfunction, you have to start watching your diet or your habits, or we're going to prescribe you some drugs to counteract the changes in your microvascular function, those predictors. Does that make sense? So we can kind of forecast the future. So the sequence of blood flow through the microvasculature goes, the terminal arteriole goes first. It's the last arteriole before the bed. Then blood enters the medarteriole, and then it enters the capillaries, not on this picture though, and thoroughfare channel. From thoroughfare channel, blood enters the postcapillary vein. Okay? That makes sense? Medarteriole and thoroughfare channel form so-called vascular shunt or anastomose. So anastomosis can be, for instance, arterial, meaning you have one artery connected to another artery. Does that make sense here? You're going to find a lot of arterial anastomosis in coronary circulation. If you remember even vaguely the cerebral circulation, the circle of Willis, the anterior and posterior communicating arteries are essentially arterial anastomosis. Posterior communicating arteries are anastomosis between the posterior cerebral and middle cerebral arteries. Does that make sense? Like circle of Willis. So posterior cerebral, middle cerebral arteries, that's anastomosis. Okay? They can be venous. Median cubital vein is a typical anastomosis. Okay? And they can be arteriovenular anastomosis, like this one, or this one, or anastomosis. So anastomosis provide an extra collateral path for the blood flow. Now, a single capillary bed can encompass from 10 to 100 individual capillaries. You can see that capillaries anastomose as well. And the blood flow through capillaries is regulated by the precapillary sphincters. The circular smooth muscle structures close and open in response to nutrient and oxygen needs of the tissue that these capillaries perfuse. I'm going to stop for a second, and I'm going to kind of discuss this with you a little bit. First, the word perfusion. Does it make sense? Perfusion is essentially a blood flow through the tissue. Tissue perfusion. Does that make sense to you? We can say total kidney perfusion, or perfusion of a small patch of a kidney, or perfusion of a small patch of a muscle. Got it? Now you have to appreciate, if tissue is hypoxic, what's going to happen to the perfusion of that tissue? So say tissue is hypoxic. What will happen in response to hypoxia? Increased perfusion, exactly. Does that make sense? Because it has low oxygen, and the perfusion will increase to deliver more oxygen. Got it? So we kind of take a sneak peek into the regulation of tissue perfusion. For instance, when you're active, your muscles require more energy and nutrients, and your muscle perfusion, blood flow to the muscles, increases dramatically. We good? And that perfusion will be regulated by precapillary sphincters. One super important statement. We just know that capillary beds are all different. And if you think about it, that makes perfect sense. Capillary beds in the kidney will be different from capillary beds in the muscle, will be different from capillary beds in the brain, and so on and so forth. Does that make sense? So, when next time you are reading a press release or a manuscript about studies in the model, say mouse model, some cardiovascular disease stuff, right? They say we studied this effects in the muscle or in the skin or in the kidney. Just keep in mind that it doesn't encompass the entire circulatory system. You understand what I'm saying? So some effects that are true in the muscle may not be reproduced in the brain and may not be reproduced in the kidneys. Am I clear? We're good? Any questions? So, blood vessels are essential regulators of the blood flow and blood pressure. So let's define each term. So what is blood flow? It's the volume of blood that goes through a certain structure in 1 minute, measured in milliliters per minute. Does that make sense? So blood flow, systemic blood flow, is equivalent to cardiac output. Does that make sense to you? So blood flow through the systemic circuit is 5.25 liters per minute. That's how much blood is moving through the entire circuit in 1 minute. Basically your cardiac output. Does that make sense? Now, we're going to do some math. Don't be scared. It should be fine. If your cardiac output, for the simplicity, I'm going to put 6 liters per minute. 6 liters per minute, okay? If your cardiac output is 6 liters per minute, first, what is your blood flow, systemic blood flow? 6 liters per minute. Now I'm telling you that blood flow through the kidneys is 25% of systemic one. What's the blood flow through the kidneys? If systemic is 6 liters, the quarter of it will be 1.5. Yeah, 1.5 liters per minute. Does that make sense? Say, cerebral blood flow is 15%. If 6 liters per minute, 0.9 liters per minute. Are we clear? So I want you to appreciate that blood flow is the volume over time. Now, blood pressure, it's a force per unit area. And that pressure is exerted on the wall of the blood vessel. We measure blood pressure in the large arteries. So, systemic blood pressure is proportional to the blood flow. If your blood flow increases, what happens to the blood pressure? Increases. If your blood flow decreases, your blood pressure decreases. If your heart stops and there is no blood flow, your blood pressure is 0. Okay? Now, what generates pressure is the resistance of the blood vessels. Your vessels do have some resistance, opposition to flow. Okay? Basically, it's a friction between the blood against vessel wall. So, we can write the following equations. Blood flow equals blood pressure over peripheral resistance. Does that make sense? Now let me ask you this. You have a patient in your possession. Do you measure blood flow? No, you don't. What do you measure? Which parameter? Blood-related parameter. Pressure. So we need to convert that equation to blood pressure-related stuff, right? So first of all, we figured that systemic blood flow equals cardiac output. So that gives us that cardiac output equals blood pressure over peripheral resistance. which gives us the most important equation for the blood pressure regulation considerations. Blood pressure is directly proportional to cardiac output and peripheral resistance. Are we clear? Let me explain what it entails. How can you increase your cardiac output? Which parameter of the heart will increase, for instance, in the exercise? Heart? No. Which cardiac parameter specifically? Heart rate. Okay, heart rate goes up. Does that make sense? Cardiac output increases, your blood pressure increases. Makes sense, right? Now, that's the way to increase blood pressure. One way to increase cardiac output somehow. How can you increase peripheral resistance? Okay, let's talk about it. In the long-term perspective, peripheral resistance will go up as your blood becomes more viscous. Increased blood viscosity. What can make it more viscous? Dehydration or increased number of red blood cells. There are conditions which people produce more red blood cells than they need. That runs the risk of, stroke and, blood pressure abnormalities. Also, when people take erythropoietin as performance enhancement drug, that also affects the blood pressure. Vessel length. The larger body size, the larger blood pressure. There are mechanisms to accommodate for it, but generally speaking, when people become bigger, not athletically bigger, that leads to increased blood pressure in the long-term perspective. Flow turbulence. This image, right above the table, shows you the laminar flow on the left and turbulent flow on the right. You can observe same features in the river. When you have a smooth river bed, the flow of water will be laminar. But if you throw a large boulder, the water will start to go around the boulder, forming these little whirlpools, and that's turbulent flow. Turbulent flow increases resistance to flow. Now, what do you think this can be in the blood vessel? A plaque, an atherosclerotic plaque. Okay. In fact, turbulent flow can be observed at any branch. When vessels merge or diverge, you can see turbulence in the flow. Does that make sense to you? Cool. However, these factors, turbulence, viscosity, body size, they are sort of long-term changes. Changes to the vasculature that can occur almost immediately, changes in the vessel diameter. Okay? That makes sense? And it turns out that the peripheral resistance here is inversely proportional to the radius of the vessel in the power of 4. Now what does that mean? Imagine that you have a blood vessel with a radius of 20. Okay? You go from 20 to 10. You decrease the radius of vessel from 20 to 10. So what's the decrease fold-wise? It's twofold, right? Turns out that the peripheral resistance will increase 2 in the power of 4, 16-fold. Does that make sense? I can do all the math with you if you want. So basically, that's R1, and then you have R2, which is 2 times less, right? So power 2 R1 over 2. Okay? So if you put it in the power of 4, it gives you 16 times higher peripheral resistance. You can easily observe it in a very, very simple way. When you go downstairs to the breakers next time, pick 2 straws. The regular straw and the coffee stirrer. And try to drink through one and then through another. The amount of effort that you should put is not proportional to that. Actually, if you ever happen to be in Houston, Texas, there's a great museum there called George McGovern Health Museum. It's really good. And they have this really simple demonstration. You have to pump fluid through the pipe. And there are 3 pipes that are progressively smaller lumen, representing normal blood vessel and blood vessel with atherosclerosis. So I had no problem pumping fluid through first one. Second one, I had to put some effort. On the third one, and I'm like, 180 pounds, I had to hang on the pump just to move it. It was so hard. Now imagine it's not me, it's your heart. So it's a real problem. Does that make sense? So in our conversation of blood pressure regulation, we're going to be focusing almost entirely on the regulation of the vessel diameter. So before we move on, we need to figure out the relationships between different parameters in the blood vessels in the systemic circuit. So first, let's talk about vessel diameter. The largest are the elastic arteries. And you can see that vessel diameter progressively decreases, reaching its minimum at capillaries. And then it starts to go up, reaching the largest veins. The biggest one of those, of course, is vena cava. Does that make sense? Now, second thing is blood pressure. So look at this. Blood pressure in the elastic veins decreases not very steeply. Can you see that? It's almost constant. But then as the blood enters muscular veins, and especially arterioles, blood pressure drops quite precipitously. This is why we call arterioles resistance vessels. So far, we good? Then look what happens. In capillaries and in veins, blood pressure, it drops, but not as much as it does in those resistant vessels. So the main drop in the blood pressure occurs in arterioles. Now what about the cross-sectional area? So again, think about the river for a second. So imagine you have the river that runs from the mountain hill here. The river that runs from the mountain hill, it's going to be a river, okay, runs from the mountain hill down into the valley. Okay, you got it? So as it goes downhill, through the really narrow bed, the flow rate is pretty high. Can you see that? Can you appreciate that? I'm going to go over it one more time. As it enters the widening in the valley, meaning capillaries, that's widening, that's a total cross-sectional area, that's the total area of all capillary lumens. Basically a lot of very tiny channels, but a lot of them. The flow rate is the slowest. And then when it enters more, sort of narrow point, the flow rate increases. So essentially, the largest cross-sectional area is in the capillaries. The flow rate is the slowest in the capillaries, fastest in the arteries. Veins kind of in the middle. Does that make sense? Now, what exactly generates the blood flow in the systemic circuit? Well, we have to consider all components of cardiovascular system to figure out how blood pressure works. So what generates flow of blood? Which organ? Heart. Look, if I'm asking you a heart question, I will announce it. Heart. So heart pumps. If there would be no resistance, would you have any pressure? No. No, you would have none. That make sense? If there is no resistance to the flow, there is no pressure. Okay? So resistance of the blood vessels generates the pressure. When blood is released from the heart into the aorta, and we're talking systemic circuit, the blood pressure is actually highest. You see systolic pressure, diastolic pressure. In the blood vessels close to the heart, the blood pressure is pulsatile. That's your word, pulse. Okay? Now you can see as the blood enters from elastic to the muscular to the arterioles, the difference in systolic versus diastolic decreases, okay, so that pulse pressure goes away, and it also drops quite greatly to about 35 millimeters of mercury in the capillaries. So recall for a second the structure of the capillary. Endothelial cells and basal lamina. They're fragile. What do you think is going to happen if your systemic blood pressure rises to some insane numbers? What's going to happen to the capillaries? You're going to get damaged. Yeah. Does that make sense? So there are mechanisms that do not allow that. But if those mechanisms, protective mechanisms, fail and blood pressure in the systemic capillaries goes above 35, there are all kinds of bad consequences that can arise from that. Does that make sense to you? Now, venous blood pressure continues to decrease, and it essentially reaches a zero. Where? It's not even vena cava. Where the blood pressure is zero. Where? Right atrium. Well, left atrium too. But we're talking systemic circuits, so right atrium. Does that make sense? Now, I want you to understand one really important and really simple thing. Blood follows the pressure gradient. Pressure is highest when ventricle pushes the blood into the aorta. Does that make sense? And then it declines all the way down from ventricle to the right atrium, from left ventricle to the right atrium. And blood will follow from the high pressure to low pressure all the time. Does that make sense? So basically, you may have heard, technically it's possible, the person-to-person blood transfusion. It's possible. But transfusion from vein to vein is not really working because the blood pressure in the veins is low everywhere. In one person it's low, in another person it's low. So you have to do actually transfusion from the artery into the vein, because then you have the proper pressure gradient so the blood can actually move. As any fluid, really. Does that make sense? Blood always follows the pressure gradient. Higher pressure gradient, higher pressure gradient, that's your pressure gradient right here, the faster the flow. Low pressure gradient, slow flow. Does that make sense? So you're all comfortable now with the concept of systolic and diastolic pressure, I believe so, right? All good. Now, a few more definitions. Pulse pressure. It's the difference between systolic and diastolic. So pulse pressure is present in elastic and large muscular arteries. Mean arterial pressure, how it's calculated. Diastolic pressure plus 1/3 of the pulse. Say somebody has 120 over 80. What's the diastolic pressure? Simple question. 80. Pulse pressure? 120 over 80. The difference is 120 minus 80 gives you what? 40. Now 1/3 of 40 is about 13. Does that make sense? 80 plus 13 gives you mean arterial pressure of 93. Just a convenient parameter to compare. Got it? Now, think about vascular anatomy. We have heart, right about here. It pushes the blood up, but is it a long way to go to the brain? Not really, right? And then to the legs, it's quite a distance, but, well, it goes down anyway, right? And then you have venous return. And blood from the head, it's kind of okay. It's going to just follow the gravity. Not a biggie. But what about blood from the legs? Think about this. Blood has to go against the gravity. And it has to rely on that residual pressure generated by the heart. So are there any other mechanisms that would ensure the return of blood through the veins to the heart? There are several. First is muscular pump. When muscles contract, they squeeze the veins, sort of milking the blood upwards. The reason why blood in your legs is going upwards, not downwards, is the presence of venular valves, just like those, semi-lunar valves between the ventricles and the great vessels. Those valves prevent the backflow of the blood. Are we clear? So, if you don't move, like if you're sitting, blood gets stagnant in your legs because there's no muscular pump. It has harder time to go back to the vena cava. Makes sense, right? Stagnant blood is more prone to thrombosis, leading to various cardiovascular events. This is why it is recommended, if you sit for a long time, from time to time, stand up, walk around, just to get that blood going. That make sense to you? Yes?
Question. When you're using the bathroom and once you're there for a while and then your legs go numb, is there a reason for them to go numb?
Say it one more time.
You know when you're using the bathroom,
okay,
once you're there for a while and
okay,
your legs start going numb, is that due to lack of blood flow?
Probably not. It's usually due to some maybe uncomfortable positioning of that presses on the nerves. It's either sciatic nerve, well, probably sciatic nerve, that will block the signal transmission and perception. That's probably not due to the blood flow. It's mostly due to the nervous blockage.
So is that also why you just crisscross applesauce for a while you're eating?
See again?
Why do you say crisscross applesauce for a while? You're
get numb? Yep. For that similar reason, yes. When you just sit for a long time, they're not going to go numb, but actually for long flights, and as far as I know, I believe I'm not sure. I know that there is a direct flight from Los Angeles to, like, Melbourne in Australia, which is like 14 hours or something, like insane time. They recommend to walk every so often, just to, huh?
They have like standing areas where you can get up and like lean your back up against while you're standing up, so like the blood goes. Because I was on an 18-hour flight from Detroit to Korea.
Oh.
And they have like it's like standing room.
Yeah. What? Yeah. Was it a civilian plane?
Yeah.
Yeah. So, well, every large transatlantic plane will have enough space for you to maybe not jog, but at least walk around. Yeah, absolutely. It's very recommended to do so. Okay. Now so this is called muscular pump. Another type of pump is called respiratory. So in respiratory pump, think what happens when you take a deep breath. Your thorax expands. Okay, expands. Your abdominal cavity actually squeezes, and that pushes the blood from the abdominal cavity into the thoracic part of the vena cava. Does that make sense? So we've got three main mechanisms that ensure the proper blood flow in the veins. It's valves, respiratory pump, and muscular pump. Now, blood flow regulation is, I don't want to say well, it's not really complicated if you consider what's the point. So first we're going to talk about the regulation of the systemic blood flow. What can change your blood pressure? Cardiac output. We're clear? Peripheral resistance. Blood volume. Now, in which situation you want to increase the blood pressure? I consider it a simple question. When do you want to increase the blood pressure? When you're exercising, to increase the blood flow. Any other? Excellent. Blood loss. When your blood pressure is low, you want to increase your blood pressure to maintain proper tissue perfusion. Does that make sense? So, sorry. That's one thing. When do you want to reduce the blood pressure? When it's high. Yeah, when it's high. Yeah, that's basically like in any case when blood pressure is abnormally high. Does that make sense? We're going to talk about what can cause the high blood pressure and stuff like that. But we're going to be focusing mostly on how to increase the blood pressure, because we're talking about otherwise healthy individuals. So, healthy individual loses blood, okay? Or healthy individual starts to exercise. So in exercise, you basically do the following. You increase the respiratory and muscle pump activity, and you stimulate sympathetic system. So increased respiratory and muscle pump increases venous return. Do you understand why? Okay, so muscles squeeze your veins harder, pushing more blood through the veins into the right atrium. Okay, increasing venous return. Your heavy respiration also contributes to higher venous return, pushing the blood towards the atrium, through the veins. That makes sense? So, respiratory and muscle pump increase venous return. That increases end diastolic volume. That increases stroke volume. Increases cardiac output. Okay? Does that make sense to you? Now, when you exercise, your sympathetic system is activated. Sympathetic activity increases the force of contraction, which decreases and systolic volume, again increasing cardiac output. Yes?
Is this really similar to the other chart that we had?
On the cardiac output?
Yeah.
Absolutely. Absolutely. So it's basically the regulation of cardiac output. But think about this. Cardiac output is not the ultimate goal, if you know what I mean. So the purpose essentially is to deliver sufficient amount of oxygen and nutrients to the tissues. How can we do it? By increasing blood flow. How can we increase blood flow? We need to increase blood pressure. How can we increase blood pressure? We can increase cardiac output. Does that make sense? So yeah, absolutely. It's virtually the same. Right? So when you exercise, you increase venous return, which increases EDV. You increases contractility, which lowers your end systolic volume, and your heart rate goes up. You can basically, except for the venous return, you can achieve the same if you just shoot epinephrine. That makes sense? That will increase the blood pressure. And this is what we do when the patient has anaphylactic shock. Injection of epinephrine will increase contractility of the cardiac muscle and increase heart rate, yeah. Increasing blood pressure. Because we're going to talk about shock, whether it's cardiogenic or hypovolemic or circulatory. Now, obviously, there's no way for us to just, I'm standing right there saying to myself, how about I'm going to increase my blood pressure and just increase it? There are mechanisms regulating it. So we're going to talk about some controls here. First is baroreceptor reflex, okay, which is somewhat shown here. So look at this. Let's talk about half of the picture. Say you have increased blood pressure. Increased blood pressure is sensed by baroreceptors, mostly in the sinuses of carotid artery and in the aortic arch. Those baroreceptors send the signal to the medulla oblongata. Okay, so it's medulla oblongata. And in case of elevated blood pressure, it will stimulate cardioinhibitory center and inhibit cardioacceleratory and vasomotor center. Does that make sense to you? When cardioinhibitory center is stimulated, heart rate and stroke volume go down. When vasomotor center is inhibited, blood vessels relax and dilate. Your blood pressure drops. Does that idea make sense? Now, in case of low blood pressure, say blood loss, baroreceptors pick up reduced stretch, stimulate cardioacceleratory and vasomotor center, that increases heart rate and contractility, increasing stroke volume, increasing cardiac output, and vasoconstriction occurs. Increased cardiac output, bless you, together with the vasoconstriction, restores the blood pressure. Does that make sense? Now, if you think about it, your blood pressure kind of fluctuates. Teeter-totter idea, okay? Now, what can be a trigger for, say, increased blood pressure? Increased blood pressure. How does elevated blood pressure change blood flow? Increases blood flow. increased blood flow, how will it change the overall tissue perfusion? It will increase it. Why do you need to perfuse tissues? What do they lack? What do they not have and want to, huh? Oxygen. Turns out, you're absolutely right, first of all. But oxygen is low oxygen is one of the triggers for increased blood pressure. But perhaps more important trigger is elevated carbon dioxide. Okay? So elevated carbon dioxide or lower pH, basically the same, will trigger elevation in the blood pressure, blood flow, and tissue perfusion. Now, what are the mechanisms? Sorry, what are the mechanisms? Same cardioacceleratory versus cardioinhibitory centers. So if you need to increase blood pressure, cardioacceleratory center will be stimulated. If you need to decrease blood pressure, cardioinhibitory. I have one more question. So, are you comfortable with the concept of cardioacceleratory versus cardioinhibitory center? Which one is under sympathetic and which one is under parasympathetic control? Very good. Cardioacceleratory is sympathetic control. Cardioinhibitory center is under parasympathetic control. We good? Clear? Now let's say you have well, if you have a constantly low blood pressure, you feel like crap. And it's really rare to have constantly, like, dangerously low blood pressure. We're going to talk about what hypertension is. But due to stress, for instance, or, well, say due to the stress, you may have a prolonged elevation in blood pressure. And that leads to baroreceptors getting used to it. So they take the new elevated blood pressure as a new normal. So how can you regulate blood pressure in this case? In this case, the long-term control is renal. Okay? And there's a direct renal control, which is super simple. Think about this. High blood pressure can be reduced by peeing out. Think elevated blood pressure means more blood gets to kidneys. Are you with me? You with me? Everybody's on board? More blood to kidneys means more urine is produced. More urine is produced and released, the less blood volume you have. The less blood volume, the lower the blood pressure. And opposite. Say you lost some blood. Like you bled. Okay, you donated blood. Okay, you donated blood. Your blood volume decreases, your blood pressure decreases. Urination goes down. Water is conserved, your blood volume and blood pressure restored. Make sense? That's called direct renal control. Go ahead.
Is that the same thing that would happen if you donate plasma too?
Anything that will reduce the blood volume. Absolutely. Now, what are the hormonal controls of blood pressure? Well, one is pretty straightforward. Short-term controls via sympathetic mediators. We talked about it a gazillion times already. Epinephrine and norepinephrine directly stimulate cardio, which center? Cardioacceleratory, and for blood vessels it's going to be vasomotor. Cardioacceleratory and vasomotor centers are stimulated by epinephrine or norepinephrine. That leads to increased heart rate, increased contractility, and vasoconstriction. That leads to elevated blood pressure. Does that make sense? Now, what about long-term kidney regulation? Kidney regulation. That's kidney. That's called sometimes indirect renal mechanism. Now, kidneys produce renin. Renin is a unique hormone. It's the hormone that is an enzyme at the same time. Renin activates angiotensin II. So here I place the picture how it works. So angiotensin is produced from angiotensinogen. Angiotensinogen, produced by a liver, is converted by renin into angiotensinogen 1. Angiotensin 1 is converted into, angiotensin 1 is converted into angiotensin 2 in the lungs by angiotensin converting enzymes, ACE. And then angiotensin 2 will stimulate thirst, so you'll drink, and it will stimulate release of aldosterone and ADH. Remind me from our glorious days of endocrine system, what's the function of antidiuretic hormone? To make you do what less? Pee. You pee less, you conserve water. What's the function of aldosterone? Retention of what it increases? Retention of water indirectly through retention of sodium. Sodium is retained, water is retained. You keep water in, blood volume goes up, homeostasis is restored. You either drink or you keep water in. Does that make sense? That's your indirect renal mechanism. Turns out you can target this mechanism to treat high blood pressure. It's quite an interesting story, actually. So, when people get bitten by a viper, one of the very characteristic signs that they develop is a precipitous drop in blood pressure. They can actually die of a shock because blood pressure tanks. And scientists ask themselves, why does it happen? And they managed to isolate the compound. The first drug was called captopril. I think there's enalapril now and a lot of drugs that are called ACE inhibitors. Turns out the venom of various vipers contain the inhibitor of angiotensin converting enzyme, which prevents angiotensin II synthesis and prevents release of aldosterone and, blood pressure drops. And the two fellas, Koshman and Ndeti, never remember the name, so I had to read it, they got the Lasker Award. It's, in terms of the prestige in medical field, it's second to Nobel. And Lasker Award is awarded to more practical developments. Like if you look at physiology and medicine prizes, sometimes they kind of not more fundamental. Okay, so they got Lasker Award for discovery of ACE inhibitors. By the way, one of the treatments to maintain a person who was bitten by viper or similar venomous snake before you get this person to the emergency room, coffee, any caffeine, to increase the heart rate and maintain blood pressure, just to make it through those 20 minutes that you drive the person to the closest ER. It's not a treat, it's not a cure, just, to maintain the heart rate. Does that make sense? Now, this, we're going to talk about that summary and take a break. This shows you sort of a general idea of how blood pressure is controlled. So first of all, you can control it through two main components. The cardiac output. Increased cardiac output means increased blood pressure. Or you can increase peripheral resistance. Now let's get the peripheral resistance out. Can you rapidly increase body size? No, unless you Hulk. Okay. Can you rapidly, in the matter of seconds, increase blood viscosity? No. Probably days. Maybe hours. Okay. Now what you can do is to change the diameter of blood vessels. Okay. Now, how can you, what do you need to activate? Which center to constrict the blood vessels? Vasomotor. That makes sense? Vasomotor center, which is under which control? Sympathetic. And sympathetic activation may be caused by stress, exercise, you all know that. Now, what about cardiac output? Basically, two ways to increase cardiac output. You can increase heart rate or stroke volume. How can you increase heart rate? You activate cardioacceleratory center in response to either low blood volume and blood pressure or abnormal blood chemistry, acidosis, hypercapnia. I'm going to introduce this term. Okay. Sorry. Hypercapnia. Hypercapnia means elevated CO2. Or, that matter, hypoxia. Does that make sense? So in response to acidosis, high CO2, low oxygen, chemoreceptors will pick up the signal, activate cardioacceleratory or vasomotor centers, and will increase heart rate. Does that make sense? Good? Now, what else? That's heart rate. What about stroke volume? You can increase stroke volume by increasing end diastolic volume, meaning increase venous return. How can you do it? Muscular or respiratory pump, or sodium and water conservation by kidneys. That's your renin, angiotensin-aldosterone system, right? That's your antidiuretic hormone. That's your, for that matter, really drinking thirst. Got it? Or, instead of, well, in addition to increasing end diastolic volume, you can decrease end systolic. How can you do it? Pump the blood stronger, which means sympathetic activation and increased contractility. Okay, so this flowchart doesn't really represent a lot of intermediate steps, but I tried to spell them out as we were going through. And you may have noticed that I didn't work my way from the stimuli to the response. I worked my way from how we can achieve a certain response. Okay? Like, how can we increase cardiac output? How can we increase peripheral resistance? Does that make sense to you? So we're going to take a break. And when we come back, we'll review the quiz. Some other fun stuff.